What is SOX2? Well, SOX2 is a transcription factor that maintains pluripotency and self-renewal in embryonic stem cells. It regulates genes involved in early embryonic development. It also controls neural progenitor cell fate, thus essential in neurogenesis. These are the various areas where SOX2 works. SOX2 is initially expressed in pluripotent founder cells of blastocyst and plays the key role in embryonic development of three cell lineages of ectoderm, endoderm, and mesoderm. You can see here mesoderm gives rise to skin and bone, endoderm gives rise to esophagus, trachea, stomach, lung, tongue. Ectoderm gives rise to mainly the neural tube, central nervous system, peripheral nervous system, inner ear, and dental epithelium. SOX2 expression in adult stem and progenitor cell is also essential for tissue homeostasis. It is also very, very important for tissue regeneration. Dysregulation of SOX2 contributes to tumorigenesis, and cancer. Now, as far as gene details are concerned for SOX2, the full name of the gene is SRY or sex determining region Y box 2. The gene family from which SOX2 belongs is a group of transcription factors with a high mobility group HMG DNA binding domain. SOX2 crosstalks with a variety of signaling pathways which you can see here that regulates proliferation, survival, and tumorigenesis as also mentioned earlier. It also includes in the pathways molecules such as EGFR, SHH, sonic hedgehog, hippo, beta-catenin, TGF-beta SMAD signaling pathways. In general, SOX2 positively or negatively regulates proliferative or anti-proliferative signaling pathways respectively, leading to enhanced proliferation, survival, and tumorigenesis. As you can see here, SOX2 is essential for proliferation. It is also useful in differentiation. It also helps in migration and invasion. It also helps in stem cell phenotypes, cellular survival, and cancer. These are the various factors which SOX2 is involved with so that it can control these various aspects of cellular growth and survival. Now, some of the developmental roles where SOX2 is involved is that it along with other factors like OCT4, NANOG, SOX2 forms a core regulatory network in embryonic stem cells. You can see in this diagram, SOX2 forms a triad. It coordinates with OCT4 NANOG to help in growth of embryonic stem cell, self-renewal, and pluripotency. It also helps, this triad also helps in differentiation of the ectoderm, mesoderm, and endoderm. It is required for eye, brain, spinal cord, and sensory organ development. It is also involved in haploinsufficiency, where only one working copy is present, and it can lead to developmental disorders like anophthalmia, esophageal genital syndrome, or AEG syndrome. SOX2 regulates various biological processes leading to oncogenic consequence. In general, SOX2 represses cell cycle upon differentiation. signals to maintain stemness and accelerate cell cycle progression upon growth signals for proliferation. SOX2 exerts an anti-apoptotic effect to promote survival of cancer cells. SOX2 also triggers EMT processes to enhance tumor invasion and metastasis. Finally, SOX2 induces cancer-specific autophagy to confer chemoresistance. Therefore, the various research and clinical importance of SOX2 are induced pluripotent stem cells, one of the Yamanaka factors, cancer, overexpressed in several cancers, example glioblastoma, lung cancer, breast cancer, etc. It promotes stemness and resistance to therapy. Marker, it is also used as a marker for neural stem cells and often used in developmental biology studies. Now, if we sum up how SOX2 is mainly involved in cancer, SOX2 overexpression or amplification is associated with poor survival of cancer patients. In certain types of cancer, such as gastric cancer, head and neck squamous cell carcinoma, and LSCC, higher SOX2 levels are associated with better patient survival. All types of cancers are here, they shown in this diagram, like glioblastoma, head and neck squamous cell carcinomas, breast cancer, hepatocellular cancer, ovarian cancer, cervical squamous cell carcinoma, laryngeal carcinoma, lung cancer, gastric cancer, pancreatic cancer, colorectal cancer, and prostate cancer. Now you can see here for breast cancer, SOX2 leads to increase in SOX2 level leads to proliferation, apoptosis, proliferation increases, apoptosis decreases, invasion increases. On the other hand, for gastric cancer, you can see here when SOX2 levels are decreasing, proliferation increases, apoptosis increases, and there is poor prognosis. Now, there are various targeting approaches for SOX2. The first one is to alter the endogenous SOX2 gene expression by direct gene targeting using the zinc finger based artificial transcription factor to generate the peptide aptamer for SOX2 targeting. This is the second one. Third one is to target the SOX2 DNA binding, thus inhibiting SOX2 transcriptional activity. The next one is to target the SOX2 via inducing immune responses. Okay. The fifth one is to use small molecular inhibitors of signaling pathways that affect SOX2, thus indirectly inhibiting the SOX2. Sixth one is to target protein ubiquitination and degradation to specifically shut down SOX2 expression. And the last one is to develop proteolysis targeting chimera, PROTAC,